Hi everyone, and welcome to the second Varsity Sci session. So if you didn't attend the first one, Varsity Sci is an open, free and grassroots symposium bringing together 13 societies from both Oxford and Cambridge to show off the best in research that's conducted at our universities, bring students closer with researchers and with research at this difficult time, and to present work from freshers to fellows of the Royal Society. So during this session, we are going to have a talk on cancer from Fatime. Unfortunately, our second speaker of the day was unable to make it. He had an unforeseen circumstance, which meant that sadly, we're just going to have one talk. But of course, after this talk, you are more than welcome to ask questions. So if you'd like to ask questions on the YouTube live stream throughout, you are more than welcome to. Also, you can ask questions on the Zoom live stream. So if you'd like to do that, please send a message to Yusing, my co-president, or to me, Christoph, and we'll definitely either ask your question or we'll set your video up once the time has come for the question and answer session. So without further ado, I'll pass over to Fatime for her talk on cancer.
Hi everyone, so my name is Fatime Qosaj and I've just entered the second year of my PhD. And the central aim of my research is to try and understand the molecular and spatial features that underpin a specific mechanism of tumor vascularization known as vascular mimicry. And we're interested in this specifically in a triple-negative breast cancer mouse model. Before I delve into tumor vascularization and what vascular mimicry is, I think it's important to first understand and highlight the complexity and diversity that's often exhibited by tumors. And so a tumor is more than just its tumor cells. There is a phenomenal array of other cells and cell types, as well as acellular structures that form the tumor. And these are generally referred to as the tumor stroma or the stromal compartment. And the stroma plays a critical role in tumor initiation and development, as well as tumor progression and a tumor's response to therapy. And some of these important cell types that are part of the stroma include the immune cells. It includes endothelial cells, which are the cells that form blood vessels. And it also includes acellular structures, such as the extracellular matrix, which essentially serves as a scaffold to support surrounding cells. And collectively, these different compartments, so the tumor cells, as well as all of these other stromal aspects of the tumor, are referred to as the tumor microenvironment, or the TME. And within the TME, we have the vasculature. And these blood perfused vessels play two essential roles. And so the first one, of course, is to ensure that the surrounding cells are receiving nutrients as well as oxygen so that they're well nourished. And the second role is to basically act as a conduit system via which tumor cells are able to escape the primary site, enter the blood, go to secondary sites, and essentially form metastatic lesions. And so you can imagine that it becomes absolutely critical for tumor cells to ensure that they have a sufficient blood supply and to ensure that their oxygen needs are met. One of the main mechanisms that has been studied, and one that is actually very important in normal physiology, is angiogenesis. And angiogenesis is a process defined by the ability of endothelial cells, so those cells I had mentioned earlier that are in the tumor stroma. So the ability of endothelial cells to form capillary blood vessels from pre-existing vasculature. In normal physiology, angiogenesis is tightly regulated by this balance between pro and anti-angiogenic factors. So the factors that either stimulate or inhibit angiogenesis. And in the context of a tumor, within what's often described as a disorganized and chaotic tumor microenvironment, this balance is often dysregulated. And what you see is an excess production of the stimulatory angiogenic factors. And this essentially activates what's referred to as an angiogenic switch, which induces endothelial cells to become migratory and proliferative, and essentially enabling them to construct new blood vessels. And in line with this idea that the TME is a tumultuous environment, tumor angiogenic derived vessels have often been described and observed to be irregular and aberrant, and they lack many of the features that are important for mature and intact blood vessels. And you can imagine that when researchers and scientists and doctors understood the importance of angiogenesis and understood that tumors potentially become reliant on such a process to ensure that their nutritional needs as well as their oxygen needs are met, you can understand how the development of therapies which target this process were developed. So these are anti-angiogenic therapies, or we often refer to them as AATs, and they target this endothelial cell angiogenic process, essentially inhibiting it and practically starving the tumor. And so these therapies initially were regarded as sort of a cure-all for solid tumors, but this initial excitement was short-lived, and lots of clinical trials actually reported that patients were not responding. And in many cases, especially in breast cancer, the patients often exhibited resistance, and this resistance was subsequently followed by tumor progression. And in many cases, this progression was more aggressive than pre-treatments. And so this prompted several questions. Of course, the first one being what is driving this resistance, and how are tumors able to ensure that they have nutrients and oxygen if their circulation is being targeted? And more directly, are there other forms of tumor vascularization that tumors are able to implement? And indeed there are. And this has often, and more recently actually, been attributed to a tumor's uncanny ability to implement mechanisms that are actually independent of angiogenesis, and therefore independent of the pathways which drive endothelial cells to create new blood vessels. And so one of these examples, or an example of such a mechanism, and really the heart of my PhD project, is vascular mimicry, or VM. And VM is an intriguing and mysterious biological process, as it describes the ability of tumor cells, which have seemingly acquired endothelial-like characteristics, to create and physically line their own blood vessels. And so in a way, I like to think of this as tumor cells, as the masquerade of tumor cells. So essentially they're tumor cells, but they resemble these endothelial cells. And it's been postulated that these VM networks are actually perfused with blood and therefore functional. And this alludes to the requirement that VM networks need to hook onto other vasculature which is perfused in order to become fluid-filled and functional. So we don't really understand how exactly this fusion happens, but what we do know is that these VM networks, similarly to tumor angiogenic vessels, are quite irregular and aberrant, and in many cases, they're often described as being embryonic-like, so suggestive of their nascent nature. And so this functional status of VM networks, coupled to this role that they may play in resistance to anti-angiogenic therapies, alludes to the contribution that VM may have in metastases. And we actually have work that was published in our lab to suggest this. And these 2 points really justify why we need to understand VM, because hopefully when we understand VM and understand how exactly this mechanism is taking place and how it sort of overrides angiogenesis, then hopefully down the line we can improve treatments essentially. And so here I just wanted to highlight what we think these, or what we've actually seen these VM networks to look like. So we have the tumor segment essentially hooking onto the an angiogenic vessel so that it can become perfused, and this basically acting as an escape route for tumor cells. And so VM, you can imagine, is a very contentious topic in tumor biology, and has been met with lots of skepticism, especially from folks in the angiogenesis field. And this is largely due to the difficulty of capturing these structures, which has led to a general lack in empirical evidence, which would suggest that VM networks are more than just artifacts of the tumor and actually do have a significant biological role. Despite this, there are several studies, and I highlight two of them that are important for my research, which have contributed a great deal to our understanding of what VM is and how it presents in tumors. And so these images here are from the first paper to coin the phrase vascular mimicry. And what we're looking at here are images that were derived using a special imaging technology. And these are sections from a uveal melanoma patient. And so upon imaging, we can see that, we can hopefully appreciate that the tumor cells, which can be identified by these little packaged circular structures, are lining what appears to be a tubular structure. And of course, to fit the current VM hypothesis that it is biologically relevant, we need to ensure that these tubular structures are indeed perfused. And so the question is, do they actually house red blood cells? And so these cells within this tubular structure, according to this paper, are indeed red blood cells, and therefore giving us more confidence that these networks are functional and are perfused and have some sort of biological importance in the tumor biology. And then the second paper that I want to highlight, which is actually lays the foundation of my PhD project, is some work that was published in my lab a few years ago. And the initial purpose of this work was to try and understand the molecular architecture of a triple negative breast cancer mouse model, which we refer to as the 41 model. And so they're interested in trying to identify different clonal populations that were driving different features of the tumor, such as features related to proliferation, features related to hypoxia, features related to blood vessel development. And during their characterization, or within this characterization, they identified two clonal populations which are actually capable of performing VM. And I've starred these two populations here. And of these two populations, only one had an enhanced metastatic proclivity, and that's this population here. And so this paper was very critical because it not only provided us with a VM model that we could work with, it also implicated VM in driving metastases and even identified several molecular markers that may underpin this mechanism. But one of the biggest caveats here is that all of the molecular analysis that was done in this paper was performed in vitro cells. And so how exactly this would translate in an in vivo context within the natural environment of a tumor remained a gray area in our lab. And in addition to this, we really don't have a set of markers or a conclusive set of markers that would allow us to confidently discern VM networks. And further, we really don't understand how exactly the tumor microenvironment, such as the different stromal compartments, present themselves or how they organize to facilitate VM. And so these significant gaps in our understanding of vascular mimicry have inspired the core idea and aim of my PhD project. And so I mainly focus on the first aim and quickly touch up on the second aim just because they're very connected. And on one hand, we want to understand how VM presents spatially and temporally. And what I mean by this is we want to understand in the context of tumor progression. So as a tumor develops from its early stages through the more advanced stages, and once it's actually gone into the metastatic stages, how does VM present? When does it actually occur? And in terms of spatial characterization, we want to understand how does the tumor microenvironment and its different components present? So how are the different cells organized in space? How are they interacting with cells that are capable of doing VM? And what does this mean for the general VM mechanism? And on the other hand, we're also interested in basically trying to boost our confidence in identifying these structures. And I'll go into more detail about this and exactly what I mean. But here we just want to make sure that the tumors we are generating have a stable VM phenotype, because this will essentially allow us to perform a more robust comparative analysis. And so with regards to the first aim, I had mentioned that we're very interested in the spatial and temporal aspect of VM. And so in order to address this question, we decided to implement a time course experiment, or sort of a time lapse, where we generate VM or tumors that are more proficient towards VM, and we collect them at different time points. And once these tumors are collected at different time points, so specifically here we collect our tumors at day 10, at day 15, as well as at day 20. So once these tumors are collected, we subject them to a number of state-of-the-art single-cell technologies. The first one is known as the 10x genomics platform, and this essentially allows us to perform single-cell RNA sequencing so that we can generate a molecular fingerprint of the VM competent cells, and hopefully via this method we're able to identify molecular markers that are important for the VM mechanism. And in addition to the molecular information and the transcriptomic profiling of VM cells, we're also interested in the imaging side of the analysis. And so we use two main technologies for this. The first one is referred to as the serial two-photon tomography, or STPT imaging. And this essentially allows us to generate a 3D rendering of our tumor samples so that we can visualize the blood vessels, we can visualize the individual tumor cells. So while we get a global image of our tumor, this imaging platform also enables you to perform single cell resolution imaging. And so we get a really in-depth analysis of our samples. And what's also really cool about the STPT instrument is that we have ours coupled to a vibrotome. So in addition to generating a 3D image, we're also able to generate histological sections at the same time. So this allows us to perform a variety of different molecular analyses on these sections within the same sample that we're also imaging with SCPT. So that's quite a powerful feature to have. And one of the main mechanisms, or rather instruments, that we use to perform our downstream analyses is imaging mass cytometry, or IMC. And this basically allows us to perform a highly multiplexed, simultaneous analysis of the spatial architecture of the tumor. So we're specifically looking at a large set of proteins that we think are important in VM. And so we're still generating the data and trying to gather it. And of course, the analysis part, one of the hardest parts is in its very early stages. So I just want to give you an idea of some of the data that we've generated so far, but I won't dwell too much on it. And so here what we're looking at are STPT images from a single sample that we generated. And I want to just bring your attention to section 49, where we're visualizing tumor cells, which are marked in green, and perfusion status, which is marked in red. So here we're trying to understand whether or not these perfused structures, which we assume to be the vasculature or blood vessels, are indeed lined by tumor cells. And if we look at section 49, not only do we see tumor cells within the vicinity of a tubular structure, but we also see them, they seem to be lining this structure here. And more importantly, this structure looks to be perfused. And to show you something that's a bit more convincing, this is a 3D volume. And we can see if we look through the stack of sections that we have, that the structure is indeed perfused and definitely has tumor cells in the vicinity and appears to have tumor cells lining it. And the reason I say lining is because many of these cells appear to be elongated. And so just to briefly give you an idea of the single cell analysis that we're doing, this was a large part of the project, very expensive and very time consuming. But the reason why we thought it was very important to implement this was because we were trying to understand and really see whether or not we could identify a rare population of the tumor cells that are capable or that are more proficient in performing VM. And in addition to that, similarly to the angiogenic switch that I had spoken about earlier, we wanted to know, well, we assume that there is a vasculogenic switch. So a point at which, or some sort of molecular feature or some sort of change in the microenvironment that activates vascular mimicry. So we know that that is present, but we wanted to identify when exactly is it happening. And that kind of justifies the reasoning behind setting up this experiment in a time course manner. And in addition to that, trying to understand how exactly the microenvironment is changing upon this activation of VM. And so I definitely will not dwell into this part of the project, especially as the single cell RNA sequencing field itself is in its very early stages, and they're sort of trying to grasp the analysis. And so obviously we are also in that same boat. But here I just wanted to show that we do have tumor cells that are expressing markers that we would expect them to express as VM competent cells. So those are markers of hypoxia, as well as some markers of endothelial cells. But the real molecular signature in identifying this is underway and will take quite a bit of time. And so finally, the second part of my project is to try and boost our confidence of identifying VM structures. And the reason why this has become so important is because we've learned that from the first part of the project, it's become quite difficult to identify VM. And obviously one of the main reasons for this is because we don't yet have a set of markers that would allow us to discern VM vessels from non-VM vessels. And in addition to that, in the first part of the project, we never introduced any sort of external perturbation or some sort of disruption that would drive the tumor to, in essence, rely on VM. And so the goal of AIM2 is to utilize some of the tools that our lab has developed and some of the knowledge that we have with interplay in order to influence the type of vascularization that the tumor takes on. And the idea being that we will be able to perform a more robust comparative analysis, and that this will allow us to actually say that the features we see and the markers that we identify are indeed directly related to vascular mimicry. And so this part of the project, we literally only started a few weeks ago, but I do have some preliminary data and some very pretty pictures to highlight the complexity of the tumor microenvironment in these VM tumors. And so here what we're looking at are IMC images. So basically we've taken a section that was generated from the STPT. We've taken a section of the tumor and we stained it with 35 to 40 different antibodies. And we're able to essentially get a spatial readout of all of those markers simultaneously. And so here we have markers for immune cells, we have markers of hypoxia. And so what we can generally appreciate is that there is an extensive infiltration of immune cells. And in addition to that, there's an extensive presence of hypoxia, which again, we know plays some important role in vascular mimicry. What this all means, it's way too early to say anything conclusive, but we definitely have lots of follow-up experiments and projects that we are going to implement that will hopefully answer some of these questions and make VM a bit less elusive. And so with that, I just want to say thank you to everyone for tuning in. And of course, thank you to my lab, especially Greg for allowing me to take on these projects and to actually work on VM, and Dario, who is my supervisor. And everyone here in BOLD has played an integral role in the projects that I'm involved in. And just thank you to the rest of the lab.
So thank you, Fatime, for her talk. So yeah, I'll be hosting the Q&A session right now. I have a question here. So could you elaborate on the method that you use to produce or to create that in vitro mouse model? Thank you.
For the second AIM?
Yeah, or like the triple negative breast cancer mouse model.
Oh, initially, so for the first part. So basically that part of that work is already published, and the idea behind that was to take the 4T1 cells and basically infect them with a lentiviral barcoded library. And this essentially allowed us to track different clonal populations. So we took those cells and they were individually plated, or rather they were plated into a microwell plate, each cell into an individual well. And the cells were allowed to proliferate and to grow. And those that were able to actually successfully grow were those 23 clones that were identified that I showed in that image. And so what they did with those 23 clones is they characterized them in a number of different ways. And one of those assays that they had used is a matrigel tube formation assay. And this is actually one of the standard methods used to identify VM. So essentially what it is matrigel is a proteinaceous solution that is supposed to mimic the natural environments of the tumor or of tissues. And so it's sort of like the extracellular matrix scaffolding that I was referencing earlier. And so if you grow VM competent cells on top of a layer of matrigel, these cells are able to form tubular structures. And so when they had performed this characterization of all 23 clones, they saw that these two specific populations were able to form these tubular structures. And in addition to that, when they injected these individual populations into mice, they did some downstream analyses, one of them being a CD31 PAS stain. And so CD31 is an endothelial marker, and a PAS stain stains for extracellular matrix and basement membrane proteins, which are important for vessels. And so what you'll find is that in VM, the stain will be negative for CD31, so it lacks the endothelial marker, but it's going to be positive for the PAS or the extracellular matrix proteins. And so they saw this with these specific clones that they had identified. And so then from there, that sort of became our VM model, at least in the lab.
Thank you. So another question is, do we know what role the molecular markers play in terms of their mechanism?
So at the moment, it's quite difficult to say because there have been several markers that have been mentioned in articles. And the problem is that these markers are also expressed in angiogenic or endothelial cells. So we're not 100% sure. There is some research implicating stem cell-like markers and their role in VM. But I think at the moment it's a bit too early to draw any concrete conclusions from the markers and what role they may be playing. But we also know that there's research that suggests that these anti-angiogenic therapies and rather resistance to these therapies drives VM. And so there are markers related to that and that mechanism, but that's another whole area that is still sort of underway, and there's a lot that's currently being done, but it still remains relatively a gray area.
Thank you. And another question, do we know what role hypoxia plays in VM in terms of a molecular pathway?
So research shows that when you treat a tumor with anti-angiogenic therapies, in many cases, this resistance that's observed, and it's actually been referred to as an evasive resistance, basically is linked to an induction of hypoxia. And so these AATs, these angiogenic therapies, seem to induce hypoxia. And if you think about it, it kind of makes sense because you're targeting the blood vessels, you're sort of destroying them. So you're reducing the amount of oxygen that is being basically delivered to these sites. And so that's going to induce hypoxia. And it's believed that cells that are resistant to this hypoxia are potentially the VM cells that are then able to actually perform VM. So hypoxia definitely plays an important role in driving vascular mimicry. And we actually have some evidence in the lab to suggest that, and actually the paper is it's been submitted and in review.
Thank you. So if there are any more questions, yes, please feel free to continue posting them. Otherwise, if you're watching the video later after we ended the live stream, feel free to comment it and we could try to pass on your questions to Fatime. Otherwise, I would like to wrap up this session today. And thank you again for joining us today. Thank you, Fatime, for your wonderful talk. I would like to remind you all about the keynote that we will have at 4:30 today. The keynote speaker is Professor Kim Nasmith. So we, as usual, will be streaming it on YouTube, and you can join on Zoom as well. So hope to see you guys at 4:30, and thank you again for joining.